Hi everyone. You should practice how you perform. So if you're going to run a race, you should practice by running. If you're going to write on an exam, you should practice by writing. So don't forget to practice writing out all the main concepts from each chapter, write out lab problems, write out the mechanisms so you can study this stuff. Just reading it and watching videos is not going to get you there when you're going to be writing on the exam. Okay. Chapter 15 is all about the immune system. This will be a little bit of a long one, so make sure to stop and take breaks when you need to. Today we're going to talk about immunity, the 7 components of innate immunity, adaptive immunity, tumors, and finally some diseases caused by the immune system. So immunity in general is all the structures and processes that provide a defense against potential pathogens. And a pathogen can be anything that's disease causing. This could be a virus, which is not living, an agent. It's going to be a bacteria, fungi, protozoans. These are all living organisms, right? So any organism or agent with the potential to cause disease is a pathogen. But not all of the things we think about, like bacteria, are pathogenic. In fact, less than 1% of all bacteria in the world cause disease. So bacteria play important ecological roles like decomposers, nitrogen fixers, and symbionts and have nothing to do with humans. In fact, we have more bacterial cells on and in us than our own human cells, and we need them. So we know when things like the gut microbiome are disturbed that we have health problems. So bacteria are critically important, and only some of them, a very small amount of them, are pathogens. So the types of innate or non-specific immunity include physical barriers, phagocytes, fever, interferons, inflammation, natural killer cells, and complement system, and we'll talk about all of these today. So these are all members of your innate immunity, your innate immune system, which is non-specific. Okay? This is not antibodies, so we'll talk about antibodies later on, but the innate immune system is not about antibodies. It's an automatic response to any invader. Okay, so the genes for innate immunity are inherited, and this allows the body to recognize the difference between self cells and foreign antigens. The first part of this immune response is the physical barriers. So these are going to be things like skin, sweat, tears, saliva, mucus, etc., all of which are physical barriers to foreign things getting into your body. So skin is going to be a really important physical factor that prevents foreign items from getting into your body. And this is renewed constantly. So new skin cells are replacing old ones. The skin you see now on your body will be gone in about a month. The epidermis is the top 18 or 23 layers of cells. And it's made of dead keratinized cells. The middle layers of skin are flat, living, and begin to produce keratin. And the dryness of the top layers of the epidermis and our continual shedding offers protection, okay? Because you can slough off those external layers of cells and prevent things from getting into your body. So that's the epidermis on top of the dermis. These are the skin cells that start out plump. And over time, they move to the top of the epidermis and flatten out as they go. So your skin is growing and pushing up from the dermis to the epidermis. And eventually the surface cells are dying to produce this layer of dead keratinized cells. Okay. Go ahead and watch this video, just the first couple minutes of it, to look at the ciliary escalator. So in tissues that have cilia like this with mucus on them, things can travel along the cilia, and we can do things like cough them out. Okay. The second part of innate immunity is the phagocytes, and we've seen some of these in lab already. Okay. So the first and the most abundant of these is called neutrophils. They're the first ones that get to any infection, any foreign particle. So the one type of white blood cell that's involved. The monocytes are macrophages and dendritic cells, and their job is to chew up and clear cellular debris. Okay. So if these TLRs are triggered, it will secrete a group of pro-inflammatory cytokines called chemokines that are pro-inflammatory and help recruit local phagocytes into the connective tissue. We also see organ-specific phagocytes, like the Langerhans of the epidermis, Kupffer cells in the liver, and microglia in the brain that help phagocytose or eat foreign particles. Okay, so here's a table of the phagocytic cells and their locations in the body. So you can go ahead and look at these and think about what they're doing there. And there's 4 main steps in phagocytosis. So the first is chemotaxis and adherence. Chemotaxis is going to be moving towards the foreign particle due to chemicals. So they sense chemicals on foreign substances and then go grab onto them, adhere to them. Then they're going to ingest and form something called a phagosome. When you finally get digestion, the phagosome fused with the lysosome inside the cell, and eventually the waste products are discarded. So these phagocytes are literally eating foreign particles. An important component of the immune system is inflammation. And so there is a detailed process for this. Okay, so let's imagine that we have tissue damage, like you get a cut on your skin. On those surfaces of things like blades or a knife, you have bacteria. So if bacteria enters the skin, particularly the dermis, you get this kind of response here. So the first thing that happens is a whole bunch of chemicals are released by the damaged cells. So these are going to be things like histamines, kinins, prostaglandins, cytokines. These are all released by the damaged cells. They're damaged both by the physical damage from the knife and the bacteria there too. Okay. A blood clot forms at that site, and you start to get an abscess. So you have to get an increased space here for these things to operate. Okay, then we get phagocytes sticking to the endothelium and squeezing between endothelial cells. So here are the phagocytes, the white blood cells in our bloodstream, entering the tissue so these blood cells become leaky, and the white blood cells leave to go into the tissue where they can phagocytose the bacteria. For repair, a scab forms and a blood clot underneath, so you can get regenerated epidermis and regenerated dermis. So those chemokines, those chemicals put out by the damaged cells, are going to attract the neutrophils and monocytes from nearby tissues. And if the infection is not contained, if they can't clear it within that area, the resident phagocytes can recruit new phagocytes from the blood. And that process is called extravasation or diapetesis. So how do they do this? How do phagocytes move from the blood site of infection? They are, again, responding to chemokines. And here they are along the capillary wall and moving and transferring from the blood vessels into the infected area. Okay. So we get a response to chemokines in the area by those phagocytic cells. We get rolling adhesion, where you have weak bonds between phagocyte surface molecules and endothelial sections. Capture, adhesion, and the diapetesis, where they actually have a squeezing between the endothelial cells in order to attack that pathogen. Okay, that's part 2, phagocytes. Part 3 of innate immunity is fever. This is a very general, non-specific response to infection. So it's a common way in which we diagnose infection, because if someone has a fever, it's a generalized response to that. Okay. It's caused by pyrogens, which are chemicals that reset the thermostat set point in the hypothalamus. So it allows increased heat production by brown adipose tissue and skeletal muscle tissue shivering, and decreases heat loss by vasoconstriction in order to reach a higher set point. So the pyrogens are those chemicals that help us reach and reset that higher set point. So why do we do this? Well, the advantages of fever are several. One is that increased temperature may kill or slow down the pathogen. We get increased proliferation and activity of T cells and B cells. We see an increased rate of tissue repair and of interferon production. So lots of important increases and advantages to fever. Over time, though, and some of the disadvantages is that it can cause dehydration, a rapid heart rate, tachycardia, and too high of fevers, really, really high, can cause delirium, brain damage, or death. So it is important to manage fevers when you're talking about infection. The fourth part of innate immunity are called interferons. These are polypeptides released from infected cells when foreign nucleic acids are detected in the cytoplasm. So this is a detection for particularly things that are alive, okay, and things that have nucleic acids in them. So alpha and beta interferons trigger nearby cells to produce virus-blocking enzymes. So it's a way for them to identify viruses and attack them. And particularly, they're attacking them by blocking virus replication and assembly. Okay, and this doesn't always work, right? We can see in the case of COVID and other viruses is that, of course, it doesn't always work. That's why we have other medical care and other systems to deal with this, but this is your innate immune response to deal with viruses. So some lymphocytes and natural killer cells release something called gamma interferon, this IFN gamma, which is useful to fight infections and cancer. And so we can use some of these innate immune system components to combat things like cancer. Our fifth part of innate immunity, local inflammation, is caused by tissue damage or foreign particles. Those resident phagocytes and mast cells release chemokines. The mast cells also release histamine to increase capillary permeability and that diapedesis we talked about a little bit earlier. The localized vasodilation to the area increases blood flow and brings more cells. So you get red, warm, painful area, inflammation, swelling, pus, right? You get all these things with inflammation, but it's really important in order to bring more phagocytes to that area. So there's a nice Khan Academy explanation here about this response. Natural killer cells are the sixth part of our innate immune response. And again, this is nonspecific. So innate immunity is nonspecific. But what they do is they destroy virus-infected cells, cancer cells, and mismatched transplant tissue. So there's been a major technological advances in using these natural killer cells in order to combat cancer. And so cells do mutate and become cancerous every day, and so they must be destroyed. So things like natural killer cells can help with that. They insert granules containing a chemical called perforin to any cell missing a self-tag MHC1. So these MHCs are how we identify self. And if something doesn't have that self-tag, they insert the granules containing perforin into them, which causes the lysis of cells. So if it recognizes a cell as not self, it puts the perforin in there. The perforin causes cells to lyse or break open, and then they're destroyed. Okay. Finally, we have this complement system that complements innate immunity. It complements the action of antibodies. Okay. It's activated by foreign carbohydrates or on pathogens, or exposure to antibodies. So this complement is actually both part of the non-specific and the specific immune system. When activated, proteins build a pore in the foreign cell. This is called the MAC, membrane attack complex. And you get an osmotic influx of water, and cells break open, so cell lysis. Other complement functions include increased phagocytosis, so more eating of those foreign cells, and inflammation. So you can start to see how it's all working together, right? To bring cells to that area, inflammation, fever, all these things are working together in order to create a generalized immune response against anything potentially infectious. Here's a picture of how these things recognize, okay? So we've got antibodies here that can bind to a bacterial membrane. The complement protein then can bind to the antibodies and help support the antibodies' function on these foreign bacteria. Here's what the MAC membrane attack complex looks like. Okay. And so you might want to take a break here. If you're ready, we can talk about adaptive or specific immunity. So all of that before was going to be a generalized response to something infectious. Now we're going to talk about specific immunity. This type of immunity is built over time as exposure to foreign antigens encourages the body to make antibodies. And this requires the lymphocytes, the T cells and B cells. And very often the adaptive and the innate overlap. Okay, so the inflammation and complement are used in both types of immunity. And you might have an initial innate response to an infection and then an adaptive response. So it's very common that you see these things working together. Okay. So as the cells of the adaptive immune system are working, the innate responses are also working to control and stop that infection. So a B cell begins as a stem cell in the bone marrow. And when they become mature, they gain receptors. So they start being able to respond to other compounds. Then they travel to lymph and blood. They're mostly housed in the lymph nodes, but they're also in the blood. And when they're introduced to an antigen, you get an activated B cell. Okay. We've got two kinds then. We've got plasma cells that make antibodies and memory cells that are there for a second exposure. Okay. So here's what this pathway looks like. You've got the antibody and the antigen. So antibodies are going to be on these cells. Here's the antigen binding to it. And now we have plasma cells creating antibodies and then memory cells being there waiting for the next possible infection. So these work by what's called antibody-mediated immunity. B cells have receptors to notice the foreign antigens. Okay, so usually these are a protein of foreign viruses, bacteria, or other allergens. After recognition of the matching antigen, the B cells divide. This is called clonal expansion. And some become plasma cells, while others become the memory cells. Okay, so each plasma cell is going to make antibodies. Each antibody has 4 polypeptide chains. 2 are heavy and 2 are light. And these antibodies are also called immunoglobulins. You might have heard about, especially with this COVID stuff, you might have heard about IgG, IgM, IgA. These are all different immunoglobulins that are antibodies. And there's a lot of different ones. You've got about 100 million trillion antibodies in your body. They attack a few million different antigens. But occasionally something new like COVID-19 comes up and we don't have the ability to recognize that yet. Okay? So variety is made by making millions of different variable regions on those H and or L chains. Antibodies are important. They cause opsonization, which enhances phagocytosis. They cause agglutination, clumping, and they activate the complement system. Okay. So there's a couple of tests we can use to test for these things. You might have heard about ELISA or Western blot. And we can use these to test for antibody presence in your blood. Okay. And essentially this Western blot, here's a picture of it, is a way for us to test whether that's present. Because you can use this mechanism to visualize whether it's present or not. Okay. So these memory cells become really important because they remember the antigen if they see it again in the future. So this is how our vaccines work, is that they rely on the memory cells then to increase activation for the future. So think about your COVID vaccine, your 2-part COVID vaccine. The first time was a primary response. Or if you had COVID, that was your primary response. Your second shot or your second exposure to that is a secondary response. It's much more rapid, potent, and a longer lasting response. Okay. The difference here is that a lot of people are reporting symptoms with the second exposure to the vaccine, but in general exposure to things, we often see no symptoms. Again, this is where it's a little bit different with COVID because people that get reinfected with COVID often do see symptoms, even though we see longer responses. Okay, so classically we see this as what's happening. The primary response comes a week or so after exposure to the antigen and increases, and then this varies widely depending on the kind of thing you're looking at, whether it's a virus or bacteria, and even the kind of virus. Whereas in secondary response, we see quick antibody increase and then a long, slow response over time. Okay. So when you're exposed to a pathogen, you've got a whole army of B and T cells that are produced. This can be through natural exposure, which is called a cold activation, or artificial, like a vaccine. And we're mostly thinking about, vaccine production has become a big topic over the last couple of years. But typically we could make vaccines for far more things than we do. So how do we choose? Well, we commonly make vaccines for diseases that have high mortality and can have lifelong effects for survivors. So sometimes people experience fever and tiredness from the activation of the immune system. So we're all seeing this with vaccines currently, is that when you get a COVID vaccine, you might experience fever or tiredness, but that's just a function of your immune system being activated. Okay. We get rare severe reactions to vaccines, and usually it's to ingredients like preservatives. And this is one reason why the mRNA vaccines, these newer ones like the Pfizer and Moderna vaccines, see less side effects, is that there's not preservatives required in the same way as has been in the past for other vaccines. Okay. So this was tried with COVID too, passive immunity, where antibodies are given to a person. So you can take antibodies created from human plasma or an animal donor and give them to someone. Doesn't always work that well, but it can. We also see this happening naturally from mother to fetus. So antibodies are small and they can cross the placenta. So if someone gets vaccinated when they're pregnant, they can pass antibodies on to the fetus. And from mother to baby with breastfeeding as well. So antibodies cross over in breast milk. The problem with this kind of immunity, though, is that it's temporary. So the antibodies will eventually be used up or worn out, and the recipient can't make more without their own immune response to the antigen. So this can be good for particularly vulnerable groups like babies and fetuses, but eventually they'll need to make their own immune response to that antigen. So the T cells are a really important component of the system. And Akiko Iwasaki is a virologist and immunologist at Yale who said you can't have a great antibody response without T cells because T cells help young B cells mature into these antibody making machines. So they're really important. They also fight on their own. So some T cells will trigger infected cells to self-destruct, and others can linger after an illness has resolved, patrolling tissues so that germs can't reestablish a foothold. Okay, and so one of the reasons why HIV is so devastating is that the virus destroys some of the body's T cells, and so it can't fight off anymore. So a T lymphocyte or T cell's job is something called cell-mediated adaptive immunity. They mature in the thymus with a hormone called thymopoietin. So you remember that with T cells are from the thymus, that's why they're called T cells. They provide specific immune protection without secreting antibodies. Okay. T cells do not interact with free antigens. The antigen must be presented to them. So either an MHC1 or MHC2 complex has to present the antigen to them. Okay. So like our B cells, the T cells begin as a stem cell in the bone marrow. They go to the thymus to mature where they gain receptors. Then they hang out in the lymph and the blood. When they're introduced to an antigen, they become sensitized or activated. And then they can become helper T cells, killer T cells, regulatory T cells, or memory T cells. So the three main types are regulatory, helper, and CD8. Regulatory T cells prevent autoimmunity. So there's a lot of work on these regulatory ones and autoimmune diseases because the misfunction or malfunction of T cells can cause autoimmune diseases. Okay. The helper T cells, they're called CD4 sometimes, are the vast majority of T cells. They become activated by antigen-presenting phagocytes. And the activated helper T cells secrete cytokines to help. They help recruit more stuff to that area. Okay. The cytotoxic T cells, CD8s, are also called killer T cells, and they detect bad cells like infected cells or cancerous cells. So again, another major mechanism we use to start developing cancer treatments. The cytotoxic T cell looks at MHC1 and releases granzymes to destroy the DNA of bad cells. Again, like our natural killer cells, they release perforin to make pores in the bad cells, and you get lysis then due to osmotic pressure in these cells. Okay, there's a picture of this. Cytotoxic C cell binding to the infected cell, perforin making the holes, infected cell is destroyed. So when we get a pathogen engulfed by a phagocyte or a B cell, the antigen is put on an MHC2 and presented. The helper T cell then recognizes this MHC2 plus the antigen and responds. And if it's a phagocyte, the helper T cell becomes activated. If it's a B cell, the already activated helper T cell will activate the B cell. So they're working together. Okay. And there's another good video from Khan Academy that talks about antigen processing. So good one to go ahead and watch and learn more about this process. Okay. So eventually our B cell becomes activated to proliferate, but you can see these helper T cells helping. They're binding to them as well. Okay. So similar but different. The natural killer cells are part of the innate immune system, the nonspecific, and they only kill cells that are missing a self-tag MHC1. Compare that to the cytotoxic T cells. They need to be activated with helper T cells. They target MHC1 with the antigen on it. So it's targeted killing. It's very specific. And you get adaptive immunity from cell-mediated immunity. Okay. So another little comparison from Professor Kernan that shows these differences here between types of T cells. So HIV stands for human immunodeficiency virus. And one of the major challenges of HIV is that it's a retrovirus. So it's an RNA-based virus, but then the DNA, when it infects, is integrated into the person's cells. And it can result in acquired immunodeficiency syndrome. So there's been a lot of progress in both prevention of HIV infection and also preventing HIV from turning into AIDS. Because AIDS, people can live with HIV for a very long time. Once it has progressed into AIDS, it can be really difficult as the person's immune system is going to be essentially non-functional. It was first declared a disease in 1981. And we talk about it in the immune system section because HIV replicates in helper T cells in the mucosa of the GI tract. Okay. And so T cell counts typically are going to be around 1,200. But if someone has AIDS, they're going to be below 200. And so, again, they have a major problem mounting immune responses against kind of generic even infections. Okay. So here's this process of HIV infecting a cell. See the virus entering the cell, reverse transcribing, meaning it's turning the RNA into DNA that is integrated then into host cells. Okay. Additionally, then the virus is going to be reproduced. And now it's hanging out in the person's cells, and it's going to be producing new cells, new viral particles through budding. So there's been a lot of technology thinking about how we might be able to kill cancerous cells using T cells and natural killer cells. Carcinogens are compounds that cause cell damage that result in uncontrolled cell division, which eventually can form a tumor. Okay. And tumors can be benign if they're slow growing and stay put, so they're not dangerous then if they're benign. But commonly we see this turning into malignant tumors that grow fast and undergo metastasis where they move around the body. So cells break free and spread, forming new tumors in new locations. And there's lots of treatments of cancers. This has increased a lot in the last few years and gotten better and better, especially as we do things like monoclonal antibodies that are used to recognize receptors on tumors. So we're using these components of the immune system in this area of immuno-oncology to increase cancer treatment. So there's a whole bunch of immune diseases, autoimmune diseases. When the immune system malfunctions, you can get some of these. There's a big list in your textbook that talks about all the different kinds of autoimmune diseases. And some of them are highly specific. But what they have in common is that you are having an immune response to your own tissues. Okay. We also see hypersensitivities or allergies as immune system malfunctions. So allergens can be things in the environment that are not pathogenic typically. So things like bee stings, mold, dust, dander, food, pollen are all things that could cause an allergic reaction, an allergic response in somebody, which would be activating their immune system. Okay, so in this case, what's happening is your allergen, whatever that happens to be, is binding to a B cell. And the plasma cell then are going to create these IgE antibodies that are going to bind to IgE receptors on mast cells. Okay. And then you get histamine and other chemicals released that cause that immune response to the allergen. Okay. So we see this happening with immediate hypersensitivity, where the plasma cells produce IgE antibodies. And we get eventually smooth muscle contraction of airways, eosinophils attract to the area, and potentially anaphylactic shock. That's why it can be really dangerous for allergies. Some people in some instances, we see delayed hypersensitivity, things like poison oak, which are not IgE mediated, but they're cell mediated T cells, lymphokines, not histamine. Okay, so an immediate hypersensitivity or an allergy is releasing histamine. The delayed one is not a histamine based response. Instead, you see a delayed symptom, and you can treat this with things like corticosteroids. Okay, so a lot in the immune system. Focus on the adaptive versus innate and what's in the book. And we'll talk about more of that in lab as well.